Hello everyone, I would first thank the organizer of the meeting to give me the opportunity to present the beginning of my PhD work, focusing on the role of Semaphorin 3A in the postnatal development of the enteric nervous system. The postnatal development of the enteric nervous system is characterized by the assembly of neurons, labeled here in red, into ganglia from 1 day after birth to 36 days. And the axons are organized in specific trajectory between the different ganglia. In correlation with this functional maturation, with this neuronal maturation, there is a functional maturation of digestive function with, for example, okay, sorry.
You can just look here.
Okay. In correlation with neuronal maturation, there is a functional maturation of digestive function with, for example, the progressive gain of spontaneous colonic motility at 36 days after birth. However, the mechanism involved in the enteric nervous system maturation are poorly understood. For the moment, only a few number of molecules have been described to be involved in the enteric nervous system maturation as adhesion molecule. And by reference to the CNS, the central nervous system, in which the neuronal circuitry formation have been well described, we decide to study the semaphorin family molecule, and more specifically the semaphorin 3A molecule, which is well described to be involved in the neuronal circuitry formation. In the central nervous system, the semaphorin 3A molecule is a secreted protein, and its signaling requires two receptors, NRP1, an obligatory receptor corresponding to the binding receptor of Sema3A, and another receptor of the plexin A family corresponding to the signaling receptor. The main described role of semaphorin in the central nervous system is semaphorin is a repulsive protein for the growing axon. In the enteric nervous system, we don't know if this molecule is expressed, and for the moment it has just been shown the expression of Sema3A in the gut of rat embryo, and some mutation of this semaphorin have been associated with the appearance of Hirschsprung disease. In the Hirschsprung patient, there is an overexpression of Sema3A in the distal colon, and more precisely in the muscle layer of the aganglionic segment. But we don't know whether Sema3A is involved as in the central nervous system in the enteric nervous system maturation. So first we have studied the expression of Sema3A and NRP1, the obligatory receptor of Sema3A in the rat distal colon at different stages of development, and then we have studied the role of Sema3A in the enteric neuron development. First, we analyze the expression of Sema3A and NRP1 by qPCR on Western blot in distal colon of rats at postnatal day 8. And we found that both Sema3A and NRP1 are expressed in the rat distal colon at mRNA level and at protein level, as shown by this Western blot. Next, we ask whether these two molecules are expressed in the myenteric plexus and in which kind of cell. So to do this, we take distal colon from rats at different stages of development, and we perform a dissection in order to obtain the myenteric plexus and still attached to the longitudinal muscle layer. And we perform immunohistofluorescence of Sema3A or NRP1 with a specific marker of neuronal cells, glial cells, and smooth muscle cells. Concerning the Sema3A in the distal colon of rats at postnatal day 8, we found that Semaphorin is here in green, is distributed in small clusters, which is consistent for secreted protein, as in the central nervous system. And we perform immunostaining, double immunostaining, in order to show the longitudinal muscle layer in the upper panel in red with an alpha SMA antibody, and to show the myenteric plexus in the lower panel with a TOUJ1 marker. And that stack from confocal microscopy indicates that Semaphorin is present in the area between the longitudinal muscle layer and the myenteric plexus. So Semaphorin is distributed throughout the tissue and is superimposed with the neuronal structure. At the same age, so at postnatal day 8, NRP1, here in green, is present in the neuron, and more precisely in axon, as shown by this immunostaining of this colocalization of TOUJ1 marker for the axon and NRP1. And in order to appreciate the spatial relationship between NRP1 and Semaphorin, we performed a double immunostaining of these two molecules, and we found that Semaphorin in green and NRP1 in red are concentrated in the myenteric ganglia, and they are close to each other. So with this data, we can suggest that Semaphorin is in good position to functionally interact with its obligatory receptor NRP1, and Semaphorin could act on the neuron during the development. To evaluate the effect of Semaphorin on the enteric neurons, we used a culture model of enteric neurons of rats, and we analyze the effects of Semaphorin on the formation of the ganglia and on the axonal complexity. To do that, we create a model of co-culture of enteric neuron with transfected cell, transfected with the Semaphorin or with EGFP as a control of transfection. And in the first part, we verified the expression and the secretion of Semaphorin by the transfected cell. So here we can see that in the cell transfected with Semaphorin, there is an expression and a secretion of Semaphorin, whereas there is no Semaphorin detected in the cos transfected with EGFP. After that, we examine if the neuron in vitro at 1 day of culture express the NRP1 receptor. So we perform an immunostaining, and we find that NRP1 is present at in the neuron, and it concentrated at the growing site of axon. So as our model was validated, we performed a co-culture of enteric neurons with transfected cos cell for 4 days, and we analyzed the neuron development. And we found that Semaphorin has no effect on the ganglion formation and the number of neurons per ganglion, as we can see with this HU labeling. In the presence of the Semaphorin, the ganglion size is the same than in the control condition. And in the presence of Semaphorin, there is a decrease of axonal complexity, as we can hear with this immunostaining. In conclusion, for the moment, we have shown the expression of Semaphorin and NRP1 in the rat distal colon at different stages of development. And while Semaphorin show punctuate distribution in the area between the muscle layer and the myenteric plexus, NRP1 is expressed in the neuron, and more particularly in the axon. And our data suggests that Semaphorin could act on the neuron development. For the moment, our working hypothesis is that Semaphorin is secreted by the muscle layer, Semaphorin could reach the myenteric plexus and act on the axonal outgrowth and restrict the trajectory of axon in the appropriate layer. And I would thank all of the team of the INSERMU 1235, and particularly the people working with me in the ENS postnatal development team. Thanks for your attention.